right, so today we are talking with Dr. Kevin Beier, an assistant professor of physiology and biophysics at University of California, Irvine School of Medicine. We are specifically discussing his paper that was recently published in May of 2022 in Cell Reports, entitled An Extended Amygdala-MidBrain Circuit Controlling Cocaine Withdrawal, Induced Anxiety, and Reinstatement. So thank you for being with us today and discussing this paper. Just to start off, could we get a little understanding of how you got into research, as well as this specific field of circuitry?
Yeah, it was a bit serendipitous. I can't say it was terribly planned. I had joined a lab that actually did retinal research in graduate school. And that led to a side project where I developed viruses that could map circuits in the brain. So there's an interesting property of particular viruses, mostly the rabies virus, that the virus actually moves from one neuron to another in a connected chain. And so scientists have learned to modify this virus to not cause the pathological and harmful symptoms that rabies has, but to harness it and be able to use it to understand how our brains are connected.
Yeah.
And so my research has taken the direction of actually using the virus to both map connectivity as it is normally, as well as how it adapts in response to experiences like exposure to drugs of abuse.
Okay. So you got into the lab and you found out that interested you?
Yeah, I come from a biophysics background, actually, so a much more sort of mathematical background. And I had joined the lab in graduate school largely because I liked the professor, basically. I liked the lab. And so I chose the environment over the projects. And it just, as I was there, this side project was able to develop. And that influenced. So I can't say I came into it with, this magnificent idea or a particular plan that I followed. It's more been following the data, following my interest as the projects arise.
Okay, yeah, I feel like I get a lot when I talk to researchers. I get a lot of like, well, I found a lab and then it was my whole life, and I was like, oh, okay. Yeah, so that's very typical, I feel like. So what is the main questions that your current lab at UC Irvine seeks to answer?
Yeah, so we have two major angles. One is to understand the vulnerability factors that contribute to neuropsychiatric disease. And so we're mostly focused on substance abuse as well as mental health disorders. And then we are also interested in understanding the triggers of neurodegenerative disorders such as Alzheimer's disease.
Okay.
So we use a variety of techniques to try to pinpoint what it is about particular individuals that predisposes them to particular outcomes. For example, while many individuals can take drugs of abuse, only about 10 to 15% of those develop what's called a substance use disorder. And so there's a large fraction of the population that ends up being resilient to it, whereas there's a fraction that is susceptible to developing substance use disorders. And it's not clear what those factors are that makes those individuals susceptible. So we're really interested in understanding what is it about those individuals in terms of the way their brains respond to drugs, in terms of the genes that are expressed in particular cell types that essentially reinforce those vulnerability factors that contribute to substance use disorders. Similar approach we use for mental illness, and then ultimately Alzheimer's.
Yeah. Sorry, I lost you for a second. So why do you study like circuitry behind these things? Why is understanding the circuitry of these disorders important and aiding in the end goal of, like, obviously treatment and prevention, which is the end goal of all these diseases, right?
Mm-hmm. Yeah, so I mean, these questions are not original, in the sense that there are a number of other groups, researchers that are interested in exploring vulnerability factors, but the way they typically go about it is different. So, for example, for identifying vulnerability factors to substance use disorder, the most commonly used method is called genome-wide association studies, whereby researchers take large populations of individuals and they identify particular genes or gene variants that are associated with those disorders. And sometimes they're common across drugs, sometimes they're different. And that's given us some, really useful insight into the genes and pathways and the biological factors that are relevant. But they also are limited in that they don't tell us anything about, for example, where those genes are important in the body, when they're important, and the pathways that they're really involved in to facilitate substance use disorders. So we're taking a different approach to that question, which is to say, we want to understand where in the brain and when these particular vulnerability factors sort of take place. And once we have that information, then we can merge that and integrate it with some of the more molecular and genetic things that other groups have been doing to try to get a more holistic understanding of how genes lead to molecules, lead to neuron communication, which leads to animal behavior, and how those things all relate to development of any one of these particular disorders.
So you guys primarily use murine models?
Yes, that's correct.
That is fair enough. So moving into the paper, what would you describe as the main and most important findings of the paper?
So we were able to identify a pathway in the brain that links both the withdrawal features of drugs of abuse as well as the reinstatement to drug-seeking behaviors. So the idea sort of that's emerged in addiction research and has become relatively well accepted is that during the initial drug taking, what guides it is reward and reinforcement. So taking the drug feels good, but after doing this repeatedly over time, the reward becomes blunted, and the individuals begin to take the drug not so much because it feels good, but because the individuals essentially feel terrible all the time. And taking the drug helps to relieve some of the negative aspects of the drug association. So that's the withdrawal. It leads to anxiety phenotypes, pain hypersensitivity, and things like that. And so we were able to identify a key brain pathway that mediates that. That is, if we shut this pathway off during drug administration, it prevents the development of these anxiety behaviors that emerge following withdrawal from repeated drug administration. But then there's a secondary phenomenon, reinstatement. It's essentially a way of modeling relapse in rodents, whereby animals essentially seek out the drug, for example, during the withdrawal phase, when they're primed with some sort of stimulus that reminds them of the drug. And so there are many ways to do this. Our lab used reinstatement of conditioned place preference. And so what we found was that if we inhibited the same pathway that was involved in the withdrawal, it actually also prevented the reinstatement of drug-seeking behavior. And so while these two phenomena have been somewhat linked, it makes sense that they might be, the same parts of the brain might be contributing. We were able to identify one pathway that actually seemed to mediate both of them. And so this provides an integrated framework by which we can begin to understand how it is that drug administration leads to these disparate phenotypes, but is driven by one single pathway.
So can you explain how you inhibited this pathway?
Yeah, so we used combination of what we call viral genetic methods. And these are related to what I was talking about before in terms of the viruses that I've worked on throughout my career. There are a lot of different viruses that scientists have engineered to do different things. So the one I talked about earlier, or just had mentioned briefly, was the rabies virus, and that's really used for pathway analysis to linking one neuron to the next. There are a variety of other viruses that we use. For example, gene therapy vectors, adeno-associated viruses. And there have been some trials in humans, for example, about their potential applications. And these are used to deliver genes to particular cells in the rodents. So we're able to use these viruses to target specific cells in the brain. And so in our case, it was particular types of dopamine cells. And then we introduced one particular gene called a DREAD. So this is inhibitory DREAD. It's a modified protein that when we administer a drug it's non-addictive drug, it actually doesn't do anything in a normal animal. But when we've delivered this DREAD molecule into these particular cell types in the brain, these particular dopamine cells, we're able to silence their activity. And so by taking one cell population essentially offline at a time, we can selectively examine what the contribution is of different cell populations to the behaviors that we're interested in.
So do you think that this type, I mean, obviously in its current form, this type of manipulation might not be feasible in humans, but do you think with some modification, these like specific pathway inhibitions can be possible?
That's certainly the goal. I mean, as I mentioned, the adeno-associated viruses have been in human trials for some time. There was a hiccup with an incident that happened over a decade ago that's really set the whole idea back for a while, but they've since been gaining traction again. For example, systemic administration of these adeno-associated viruses to deliver genes to patients with particular genetic deficiencies. So this isn't quite ready for prime time in humans. I mean, the idea of delivering viruses to particular sites in the brain, sounds a little Frankensteinian. And so we're not there yet in terms of the ability and sort of the ethical considerations that would come with modifying the brain in that way. But practically speaking, there's no reason it couldn't work in the sense that people have used these modified forms of viruses, for example, in non-human primates. And gotten them to work in similar ways. So the main issues would be a sort of biosafety, making sure that these things are really healthy, well tolerated in humans, in this particular application. And then the ethical concerns about modifying people's brains, that was sort of a larger issue that has been on the minds of neuroscientists and anybody involved in scientific ethics for some time.
Yeah, that's fair. So obviously this was all conducted in mouse models. How conserved are the neural circuits that you're describing in addiction and relapse, not just the one in this paper, but other neural circuits in human people?
Yeah, so all the core circuits are conserved. So mouse brain certainly is not a human brain, and that's always important to consider. That we find these pathways, we find these potential therapeutic targets, and then translating them to humans is not always trivial. So, I mean, there are a couple of things I could say, and one to sort of highlight on the last question you asked. The sort of idea with the rodents research is that it gives you pathways, it gives you areas to target, and then hopefully you can develop some sort of therapeutics that might be tolerable in humans. For example, this potential, more applicable avenue of our research, or what might come after it, would be if we've identified this pathway, maybe we can identify small molecules that can alter the pathway, and then those small molecules, are certainly more amenable to human application than would be sort of direct brain modifications. But to sort of get back to this particular question, the rodent brains are certainly different, but all the core regions are conserved. So with humans, the major difference is there's been a massive expansion of a region called the cerebral cortex. And so most of the sort of corresponding regions exist in mice. There's some controversy about what exactly the prefrontal cortex is and how it relates in humans and mice. But by and large, the same regions exist. The connectivity between different regions is fairly similar between both humans and rodents. And so, in broad strokes, where the rodent literature is often consistent with the human literature in terms of what these regions are contributing to. And for example, the regions we identified, the bed nucleus of the stria terminalis, the amygdala, these are both known to participate in substance abuse in humans as well. So I would expect there to be some relevance to humans.
Yeah. So in this circuitry, it seems like they're either overlaps with or is a very similar circuitry to what is seen in something like PTSD, where you're inciting like a lot of the same amygdala functions. Is that the case with a lot of addiction and relapse, or is this a specific pathway that just overlaps?
I mean, I think there's a lot of commonalities between different mental illnesses. So, I mean, in terms of like what the triggers might be, that would certainly be different. For addiction, I mean, it's a lot of, I mean, it'd be drug associated cues versus in PTSD. It could be a whole variety of cues, but the idea is sort of similar in that, that there are various environmental cues that have been the individual has associated with particular outcomes. And so in the case of PTSD, it's a vision, it's a sound, it's something that conjures up images, that conjure up memories. And that leads to the state of PTSD. In the case of substance abuse and drug-seeking behaviors, for example, during withdrawal when people are trying to remain abstinent, it's sort of similar things, and that certain places, certain associations trigger the memory of drug taking that then compels individuals to continue seeking the drug. And so that's actually one of the biggest issues to treating substance abuse, is to keep individuals abstinent, despite the fact that they're bombarded by drug-associated cues in their environment.
Yeah. Okay, that makes sense. So my final question is more of a zoom out version. In your field, what is the research you're most excited about that's being studied currently, and what do you think would be exciting research that needs more research, that isn't being looked at as much?
So we're most excited about, again, identifying both these vulnerability factors to substance use disorders and using sort of large-scale approaches to understand how it is that the brain actually, sort of encodes the state of drug seeking or withdrawal. So we're trying to understand basically what it is on the brain in a network level that defines sort of the drug reward state versus the drug withdrawal state versus the drug seeking state. And we're using computational approaches to try to identify very particular areas of the brain that serve as sort of master nodes, master regulators of these so-called brain state transitions. And so we're particularly interested in identifying what those are in the context of opioid abuse, so we can understand, when people take opioids, for example, prescription opioids for pain, what the triggers are that then can facilitate dependence, and that can ultimately lead to addiction. And so I think, that the kind of work that needs to be done, really sort of integration of a variety of sort of different approaches and methodologies, for example, in substance abuse research. There's a lot of really interesting research that goes into drug-seeking behavior, that goes into understanding, how we can best model addiction in rodents, and how we can understand, what genes and molecules play a role. But I think if we want to understand more holistically how addiction works, how we can model it, how we can develop therapeutics, I think it requires a crosstalk between different labs, between different researchers who have particular expertises, so that people can begin to answer questions that are sort of the low-hanging fruit that we have really not been able to be addressed by sort of a single approach or technology.
Yeah, well, I think that like collaborations in science is something that has been starting, and I've been very excited about that as well. So I agree on that front. Well, thank you so much for talking with us. It's been very interesting.